first lecture in the metabolic series, so we're going to be doing basically nothing but pathways from here on out. We're going to start with a very simple pathway that only has two enzymes. So as promised, here's the metabolic map, and right in here is where we'll be exploring today. Now this will also overlap with what we're going to explore next, which will be glycolysis, but for now we're just up in this little corner here. So your goals for this lecture are to understand glycogen, anabolic, and catabolic pathways. I'm going to put some emphasis on the catabolic system, mostly because the anabolic system is pretty straightforward and not terribly interesting, whereas the catabolic system has a couple of enzymes, one of which is extremely complicated. The entire key to all of this is to be able to wrap your head around how the main enzyme of glycogen catabolism is regulated and functions, because otherwise the reaction is very, very simple. We're going to look at glycogen metabolism extensively. Now I'm going to bring in G proteins, and if you're not familiar with G proteins, I do have a lecture available for those. Not that I'm going to cover them heavily in this class, but that it will help you understand a little bit more of how a G protein works, especially with regard to regulation of glycogen. And of course, we'll look at some associated diseases of glycogen metabolism. So glycogen metabolism is going to require quite a few different enzymes for degradation. Now we're really going to be focusing on two, but there are a host of other enzymes that deal with the different aspects of a glycogen molecule. Our key controlling enzyme is going to be glycogen phosphorylase. Okay, this is just shortened to phosphorylase. This is an allosteric enzyme. It recognizes a whole host of different small molecules, including ATP and AMP, and will respond to signals from epinephrine and insulin. But we also want to bear in mind two major tissues while we're dealing with phosphorylase. Phosphorylase is found in pretty much anything that has glycogen, but there's two key tissues, and those are the muscle and the liver. And the way to start to understand how phosphorylase works is to realize that the muscle and the liver have different roles. Okay, so the muscle is all about contraction and mechanical motion, and one of the major roles of the liver is actually maintenance of blood sugar levels. So that's going to be crucial. So kind of keep these two tissue types separate and realize that the way phosphorylase behaves depends on the tissue where you are examining it. So here's glycogen. It's just a big old blob of sugar. It has a core protein called glycogen sitting in the middle that acts as a primer for building the glycogen molecule. And it comes off in long chains of just glucose. That's all this is. Glucose, 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 glucose. Just glucose in a few different configurations. So first of all, we just have long chains. And these are connected by alpha-1,4 linkages. And we can have branches that are connected by alpha-1,6 linkages. That's not terribly important, but realize we're going to have some issues given the branching and spurs and longer chains. However, at the end of every glycogen chain is what's called a non-reducing end of glucose and a free hydroxyl. So the free hydroxyl ends. This is where phosphorylase does its action. It's literally going to nibble off one glucose, one glucose, one glucose, and one glucose. So it's just going to nibble that off. Now, as I was mentioning, we have two major sites for glycogen degradation, catabolism, and anabolism. That's the liver and the muscle. Okay, both of these extensively use glycogen. For the liver, we're talking about maintaining blood glucose levels. It has the highest concentration of glycogen by weight, and we're going to release glucose between fed states. All right, so during fasting periods, phosphorylase is going to be active, and we'll be nibbling off the ends of these glycogen molecules, releasing glucose ultimately to be released to the blood. Now in the muscle, one of the primary sources of energy in the muscle is glycogen. It has the largest amount of glycogen, not as opposed to highest concentration of glycogen by weight, but the largest amount. and muscle cells will deplete glycogen, will result in exhaustion of that muscle. So what we're going to see is the liver is actually using glycogen to maintain blood glucose levels. The muscle can use glycogen for its own activity and import glucose as well. And that's going to be very important for glycolysis. So what is the action of phosphorylase? Well, phosphorylase cuts glycogen, and essentially it's going to take some glycogen chain and residues. Here we're just showing two, and it's going to use a phosphate. Okay, it's actually going to use a phosphoric acid molecule, shown here, to release glucose 1-phosphate. Okay, and of course the remaining glycogen, which would be N-1 residues. So that's all it does, is it nibbles off one last, the terminal glucose molecule, creating glucose 1-phosphate. And this is done by the enzyme phosphorylase. Now notice this is not a hydrolytic reaction, this is a phosphorylytic reaction. It does not require ATP, it's using phosphoric acid, not ATP. That's a very important idea because we do have free phosphate in the cell, and this reaction is going to use some of that without touching the ATP supply. So if you were to do this hydrolytically, it would require ATP to phosphorylate the glucose moiety. So instead of using hydrolytic cutting, we're going to use phosphorylytic cutting and do both actions in one time. So instead of having to consume an ATP to then phosphorylate the substrate, we're just going to use phosphoric acid, just a free phosphate group, in order to cut the chain and phosphorylate the target at the same time. This is important because this bypasses hexokinase. Now, we haven't seen hexokinase yet. It's the first canonical step in glycolysis, but it can be bypassed. So hexokinase actually does require ATP, but phosphorylase doesn't require ATP. So this is a way we can get into glycolysis without using ATP. Now, it's important to note there are no transporters for the glucose 1-phosphate molecule in cells. We don't move this material around, so the glucose 1-phosphate is stuck in the cell. So we're going to have a bit of a problem here if we want to move a glucose molecule from the liver into the blood, because phosphorylase creates glucose 1-phosphate, not glucose. And as I also mentioned, there are a couple of other enzymes we need to consider. Phosphorylase, of course, is just nibbling off the ends. Shown here is these red glucose 1-phosphate molecules that are being released. And then a transferase can simply move short branches onto longer chains, so the phosphorylase could keep cutting these down. And then alpha-1,6-glucosidase will remove these little spurs that appear. Beyond that, we're not worried about it. Now, we have glucose 1-phosphate, and we need to convert this to glucose 6-phosphate. This is actually the reaction that's going to get us into glycolysis, because our product is glucose 6-phosphate, which is the first substrate, the first metabolite in glycolysis. Well, after glucose, obviously. So how does this happen? Glucose 1-phosphate to glucose 6-phosphate. Well, it turns out the enzyme phosphoglucomutase, remember mutases are enzymes that simply move phosphate groups around, will do this activity. So it has a serine that is phosphorylated, and it will attach that serine to the 6th position on the glucose molecules. So we go from glucose 1-phosphate to glucose 1,6-bisphosphate, and then that same serine will pull the 1-position phosphate off. So we go from glucose 1-phosphate, that was made by phosphorylase. The phosphomutase creates glucose 1,6-bisphosphate briefly, and then removes the 1-position phosphate, resulting in glucose 6-phosphate. This is the molecule that will go into glycolysis, or, as we can see, into gluconeogenesis, and that's going to be key for the liver. So in the liver, we need to maintain blood sugar. As I said, this is going to be the big role of the liver in this discussion. And so we have now glucose 6-phosphate in the liver, but that's not glucose. Glucose is what we can transport out, not glucose 6-phosphate. And so the liver possesses this enzyme, glucose 6-phosphatase. Okay, not a phosphamutase or anything. This is a phosphatase. And if we recall, kinases add phosphates to targets, usually via ATP, and phosphatases remove phosphates from targets. This is the same enzyme we'll see again in gluconeogenesis. We do not have this enzyme in the muscle. It is not there. And so we can use this phosphatase to convert glucose 6-phosphate to glucose. Glucose can now be transported out of the liver and into the blood. The muscle will not have this enzyme. The muscle is simply going to import glucose and convert it to glucose 6-phosphate via glucokinase or hexokinase for glycolysis, but it cannot transport the glucose back out because it lacks the phosphatase. So in this way, the liver releases glucose to the blood, and the muscle picks up glucose from the blood and converts it to glucose 6-phosphate via hexokinase, keeping it locked in the muscle cell. So that's all very well and good, and as you can see, there's only really two enzymes we're worrying about, phosphorylase and the phosphoglucomutase. But what we really need to concern ourselves with at this point is regulation. Glycogen is a limited resource, and it takes time and energy to build it back up. So we need to carefully control how we're degrading this material. So what we're talking about with glycogen degradation is control of phosphorylase, and it is an allosteric enzyme, and it recognizes energy states, it recognizes hormones, and the way it works depends on the tissue where you find it. So let's start with the muscle, because the muscle is actually the slightly more complicated form. Now, what is true of phosphorylase is, and this, by the way, doesn't matter muscle or liver, but that's true for both, there are two forms, a phosphorylated form and a non-phosphorylated form. So we can call it phosphorylase phosphate or just phosphorylase. Okay? Two forms. A is what we give the term to the phosphorylated form. So if phosphorylase is phosphorylated, it is termed phosphorylase A. The A form is usually active. It's going to take on what's called an R state or R form. We'll get to that in a moment. All you need to know right at this moment in time is the A form of phosphorylase is the phosphorylated form, and it is usually found in the R state. If phosphorylase is not phosphorylated, it is found in the B form. Okay? The B form is usually inactive and is called T. So the B form is the non-phosphorylated form, and it is usually inactive and usually found in the T state. Now, each of these A and B can either be inactive or active, but they will favor one or the other, as we're going to see in a moment. So this diagram seems very complex, and it's not that bad. We're going to break it down step by step. So on this side of the diagram, we have phosphorylase B. Notice it is not phosphorylated. On this side of the diagram, the right-hand side, we have phosphorylase A. Notice that it is phosphorylated. Okay, so phosphorylated phosphorylase is called phosphorylase A. Non-phosphorylated phosphorylase is called phosphorylase B. So if you look carefully, you will see there are equilibria arrows. One here in the phosphorylase B column and one here in the phosphorylase A column. The A form prefers the R state, and that's given by this much larger equilibrium arrow. So while the A form can take the R or T state, it prefers the R, and the R is what is active. So this is a fully active molecule of phosphorylase. Phosphorylase A in the R state. Phosphorylase B, on the other hand, if we look at the equilibrium arrow, shows a very strong favor to T. So phosphorylase B favors the T state. And notice the T state is inactive. And one way that's being shown is we see occlusion of the active site. Okay, so we see this disordered, well, not disordered, but a blocked active site. So phosphorylase B prefers the T state. Phosphorylase A prefers the R state, but they can be moved, shown by these equilibria, into the alternative state, R or T. To go between phosphorylase B and phosphorylase A requires phosphorylation. Since it's going to take phosphorylation, the enzyme that will do it is probably a kinase. Notice when we are in the T state, the active site is blocked. Whether or not it's phosphorylase A or B, the activity of the enzyme is dependent upon access to the active site. The phosphorylated form, phosphorylase A, prefers, given by this equilibrium, the R state. Phosphorylase B, the non-phosphorylated form, prefers the T state. So since we have these different states and we know their relative activity, we can talk about how those states are controlled. So let's look at the B form. When you're at rest, most of the phosphorylase in your muscle is in the B form. Now normally that means you're not breaking down glycogen, but B can be stabilized to an active state, the R state. How does it do this? Well, first of all, if ATP gets too low, that means you have a lot of AMP in the muscle cell, right? So if we go from ATP to AMP as it gets used up, AMP becomes more abundant. This is a low energy state. It will bind to a particular allosteric site on the B form of phosphorylase in the muscle, and which state do you think it's going to stabilize? Will it stabilize the inactive or the active? Take a moment to think about it. It stabilizes the R state. The R state is favored for activity when energy is low. So we need to use phosphorylase to break down some glycogen, to run it through glycolysis to make ATP. So if AMP levels are high, the B form of phosphorylase in the muscle will be stabilized to the R state by the AMP. Now let's look at the other side, ATP. So suppose we have a lot of ATP, it's abundant in the cell. This is considered a high energy state. ATP competes with AMP for the nucleotide binding site, and can you guess which state it will stabilize? Well, since we are high energy now, we don't need to make a whole bunch of ATP, and so phosphorylase would now be stabilized in the T or inactive state, which it favors anyway. Well, let's think about one more thing. What about glucose 6-phosphate? So now this can act through feedback inhibition. Glucose 6-phosphate is a downstream metabolite of glycogen degradation. If we have an abundance of glucose 6-phosphate, what do you think happens to the B form? It stabilizes again in the T state, because if we have abundant glucose 6-phosphate, we do not need phosphorylase making more glucose 1-phosphate to be converted to glucose 6-phosphate. Again, we're fine. We don't need any more material. But that's not all that the muscle does. Occasionally we have an epinephrine event. So this is very, very important for the muscle, and we're going to also have strong muscle contraction rather than just sort of maintenance muscle contraction. So in this case, the B form of phosphorylase becomes phosphorylated, turning it into the A form. The A form is not going to respond to AMP, ATP, or glucose 6-phosphate. Remember, epinephrine is a panic signal. It's the fight or flight response. And so the muscle right now, it just doesn't care about, are we overcharged, undercharged, do we have too much glucose 6-phosphate? All it cares about is we need to make energy as fast as possible for as much contraction as possible. The enzyme that will do this transfer is, as you may have guessed, phosphorylase kinase. So phosphorylase kinase will convert phosphorylase B into phosphorylase A. So what is the result of this? Well, you get tons of activity of phosphorylase. All kinds of activity, which makes sense because we're under a fight or flight response. And the rate of degradation of glycogen will rapidly increase. Also, remember, there is no glucose 6-phosphatase in the muscle cells. Do you remember why? Because glucose 6-phosphatase converts glucose 6-phosphate to glucose, which is necessary for the liver for the maintenance of blood sugar, but is not necessary for cells. In fact, they don't want to do this because they want to bring in the glucose and trap it as glucose 6-phosphate. So now that we've done the muscle, we'll do the easier version of the liver. Now, the liver pretty much is always working on anabolic creation of glycolysis or catabolic degradation of glycolysis. And you're more frequently fasting than you are fed. So the goal of the liver is to create glucose for export to other tissues. If we have glucose from the diet, we don't need to mobilize glycogen, but we have the primary form of phosphorylase is phosphorylase A. And remember, phosphorylase A is the phosphorylated form, but this is without a signal. And this is the active. This favors activity. But phosphorylase A is sensitive to levels of glucose. So if we look at phosphorylase A, it will favor the R state on its own, but the presence of glucose will drive its configuration to the T state and hold it there. So if glucose is high, either from diet or from gluconeogenesis or just previous glycogenolysis, then phosphorylase A can in fact be slowed down because of overabundance of glucose. That's pretty much it for the liver. Now, let's talk about a disease that comes with glycogenolysis, and that's phosphorylase deficiency. So what happens to individuals who don't have phosphorylase? This is primarily in the liver, where we have an isozyme deficiency, and it's titled HERS disease. Symptoms: enlargement of the liver, or also known as hepatomegaly. This makes sense because we're going to be building glycogen, but we're not going to be able to clear glycogen and release it as glucose. And since we can't clear glycogen to make glucose for release to the blood, we should expect these individuals to have hypoglycemia. If the enlargement of the liver becomes too great, or the particular case of HERS disease is extreme, liver damage is certainly to be expected. The other clinical issues are going to vary, and they range from undetectable to liver damage, growth retardation. This is going to depend on the particular patient. Again, this is a childhood disease, and this disease improves with age, suggesting that there might be alternative forms of phosphorylase as an individual ages, such as there are with hemoglobin. So now that we know sort of the general regulation of phosphorylase, is there hormonal control? And yes, there is. And they come under glucagon and epinephrine. So glucagon is a hormone that's active when you have not eaten recently, and epinephrine, as we already discussed, is the fight or flight response. So glucagon activates an enzyme called protein kinase A, and that's in a G protein system. We don't really need to go into that heavily. And glucagon and epinephrine activate liver phosphorylase, whereas epinephrine is specific to muscle. So we can have glucagon and epinephrine both promoting glycogen breakdown to glucose and lactate formation to glucose, so that's glycogenolysis and gluconeogenesis, which we'll get to later, and release glucose to the blood. Of course, if we have not been fed recently, that's where glucagon comes into play, or if under fight or flight, that's where epinephrine comes into play. That glucose will be picked up by the muscle cell, and if there's an epinephrine, we will also see glycogen activation. Overall, and I'm not going to dwell on this too heavily, the hormone, epinephrine or glucagon, activates a G protein coupled receptor system. The G protein activates adenylyl cyclase, which converts ATP to cyclic AMP. That activates protein kinase A to its active form, and protein kinase A phosphorylates phosphorylase kinase, which becomes active, and phosphorylase kinase phosphorylates phosphorylase B, making it phosphorylase A, and remember phosphorylase A prefers to be active. So now we've looked at catabolism, let's briefly look at synthesis. Now, there are multiple pathways for synthesis and degradation, so this is just one of them, and this is a very, very common theme in metabolism, to have multiple pathways doing things. In synthesis, we're going to rely on uridine diphosphate glucose, so this is just a carrier. It's kind of like the CoA part of acetyl-CoA. So you see we have this nucleotide, diphosphate, with a glucose molecule on it. It's just an activated carrier. An anabolism, which is the creation of biomolecules, often requires activated characters. We'll see NADPH, acetyl-CoA, UDP, there's a few others. The enzyme is glycogen synthase. This is our key regulatory enzyme, and all it literally does is move glucose from the carrier, uridine diphosphate, onto the end of a glycogen chain. That's it. Glycogen synthase uses UDP to move glucose from UDP glucose to glycogen. There's a small protein in the core called glycogenin. This is our primer. There are glucosyl moieties on it that act as anchors for building the glycogen chain. So here we have our alpha-1,4 linkage, or glycogenin would be here in our glycogen chain, and then UDP glucose plus glycogen synthase is just going to add glucose molecules to the end of the growing chain. There is an enzyme, amazingly named branching enzyme, that will move the chain around a little bit to create branches so that the glycogen molecule can be compacted. So glycogen synthase will continue to grow from each of these ends, and there will be more branching and more growth and more branching, more growth. How is glycogen synthase regulated? It also has two forms, A and B, where A is the active form and B is the inactive form. However, in this case, different from phosphorylase, phosphorylation inactivates the enzyme. Okay? So B is the phosphorylated form of glycogen synthase, and it is inactive, where A is the non-phosphorylated form of glycogen synthase, and it is active. And interestingly enough, this event is controlled by the same signal cascade initiated by epinephrine and glucagon. That makes sense. If we need to degrade glycogen, we don't want an enzyme also trying to build glycogen at the same time. And so the same enzyme, protein kinase A, phosphorylates phosphorylase kinase. Remember, phosphorylase kinase activates phosphorylase, but protein kinase A directly phosphorylates glycogen synthase, making it the inactive glycogen synthase form. So the same signal, glucagon or epinephrine, simultaneously activates glycogen degradation through phosphorylase and blocks glycogen synthesis through glycogen synthase. So phosphorylase A, on the other hand, as we know, is inactivated by glucose, and so glycogen breakdown ceases. Insulin activates glycogen synthesis. So here's how it works. If your blood glucose is low, phosphorylase activity will be high, degrading glycogen to release glucose to the blood. As glycogen is degraded, blood glucose levels increase, and increasing glucose results in decreased activity of phosphorylase A, because phosphorylase A is sensitive to glucose. If blood glucose levels are high, insulin is released for sugar uptake, and glycogen synthase is activated. So here's what we should see. If we look at enzymatic activity, of the phosphorylase in the synthase and its relative state A to B, and we add glucose, suddenly phosphorylase activity will be inhibited within a couple of minutes because phosphorylase A is sensitive to glucose, and glycogen synthase activity will increase and it will become dephosphorylated. Just to show you by way of signaling, this isn't a huge deal, but we have insulin interacting with a receptor tyrosine kinase, which allows activation of protein kinases, which phosphorylate glycogen synthase kinase to block its activity, which allows glycogen synthase to become non-phosphorylated and therefore active. So speaking of blood glucose, let's talk a little bit about diabetes mellitus. So this could be caused by a couple of different things. The symptoms still basically the same. Overproduction of glucose by the liver and underutilization of glucose by other tissues. Either or both. Type 1 is an actual autoimmune disease, and this will manifest around age 20 and result in loss of pancreatic B cells that produce insulin. So these individuals need insulin. Type 2, on the other hand, is adult onset. We're going to talk about type 2 a little bit more later on. Insulin resistance. These individuals need ways to clear glucose from the blood because they have actually developed a resistance to insulin. Type 1, okay, so we have entry of glucose into adipose tissue is impaired, and so the liver remains gluconeogenic. You're in biochemical starvation because you're not getting the glucose to where you need it, and because you do not have the insulin to pull the glucose into cells. You can also run into fructose 2,6-bisphosphate deficiency, and you have inhibition of glycolysis and activation of gluconeogenesis. So the other problem is, if this wasn't enough, blood glucose will be excreted via the urine and is going to take a ton of water with it. Now the flip side of that, okay, Ivan-Gerke disease. Now this is a blood glucose deficiency, not a blood glucose overabundance. In this case, these individuals do not have glucose 6-phosphatase in the liver. Remember, we need glucose 6-phosphatase in the liver to convert glucose 6-phosphate to glucose. Symptoms are very similar to what we've seen before. Hypoglycemia, because they don't have glucose in the blood. Hepatomegaly, because they're not properly breaking down the materials in the liver, which can lead to liver cancer in this case. Failure to thrive, and threat of lactic acidosis, because a lot of systems are going to be running on anaerobic glycolysis. This is a lethal disease at infancy. It can be treated. It's got to be caught. The lethality comes from the hypoglycemia and the acidosis. So to treat it, these children need a starch and glucose enriched diet, and they are going to need sugar every 3 to 4 hours. That's going to be a great deal of sugar, and it needs to be glucose and starch. Okay, we don't want to get into fructose or any of the other sugars. It's got to be glucose or starch. With proper treatment, life expectancy and quality of life is normal. And so there you have it. Here's some problems suggested this is from the Tomasco's, a short course. The chapter numbers may change a little bit, but go ahead and really work on wrapping your head around phosphorylase. That's the tough one. If you can work out the 4 different states and how they're stabilized and where you're going to find them and under what conditions, you are well on your way to understanding glycogen metabolism.